Hola, muy buenos días a todos. Mi nombre es Luis González y quiero darles la bienvenida a este seminario, An Overview of Environmental Pollutants and Monitoring Methods, que será impartido por nuestro colega Hank Han. Bueno, les platico un poco acerca de Hank. Él actualmente es el especialista en ventas senior en OI Analytical y se encuentra en Texas, en College Station. Tiene más de 31 años de experiencia en instrumentación analítica, incluyendo instrumentos como cromatógrafos de gases, analizadores de carbono orgánico total y análisis químicos automatizados, que será en lo que nos estaremos enfocando en esta presentación. Bueno, debido a su experiencia, pues él puede diseñar, puede ofrecer soluciones para una gran variedad de aplicaciones en numerosas industrias. Hank, how are you? Good morning.
Good morning.
Thank you. Well, I'm just giving a little presentation about you to the audience. So you can start now with the presentation if you can. Y bueno, entonces vamos a comenzar con la presentación. Ahorita hay que recordar que la presentación va a ser en inglés. Y bueno, debido a esto, de preferencia, las preguntas que tengan pueden hacerlas llegar en español o en inglés, no hay ningún problema, pueden ser ambos idiomas. Lo que sí les pedimos es que por favor las hagan llegar al chat y a la sección de preguntas y respuestas. Les vamos a dar respuesta al final de la presentación. Entonces, bueno, por lo pronto vamos a comenzar. Hank, you can start, please.
Okay. Good morning again, everyone. Thank you for joining us and the opportunity to present this to you today. I'll start with our applications, method and technology. So our agenda is why do environmental pollutants need to be monitored? The most common environmental pollutants, go over methods to monitor pollutants, the YSI xylem technology that we offer, and then also close up again with the available methods. So why is there monitoring? There's two types of kind of like labs that work with. There's the environmental labs, and they are the commercial labs that analyze samples for a profit. There's then your municipal laboratories, state government labs, that analyze for process control and to satisfy specific permits. Then you also have industrial labs that do that to satisfy permits as well. So instead of farming those samples out to other people to run them, they'll bring the lab into the facility and do the analysis there themselves. They also find that a little easier, as well as they're not waiting on a lab to provide the results. We've had success here in the States with facilities that can monitor their runoff or their waste streams immediately and know if they have to retreat that or then to release it, and they don't have to wait on somebody. So who does this testing? There's laboratory chains, there's independent laboratories. You've got federal, city, state labs. You have the industrial, petrochemical, chemical labs. Also, you've got universities doing research, also writing grants. Really big is nitrate, phosphorus with these grants and such, especially with global warming. Then there's also mining and plating. Really big in the United States as well, were USGS geological surveys, USDA facilities, and they were former Latchet users. So Latchet is a product that has been discontinued that was a flow injection analyzer that our instruments can be used in replacement for those. There's regulatory agencies, and some of those in depth is the Safe Drinking Water Act, the Clean Water Act as well. They developed methods for the analysis of drinking water and wastewater. They can be used to establish whether the water is safe to drink or if an industrial municipal effluent is in compliance with a specific permit, and they can release that back into the water stream. There's also Protection of Environment Part 136 and also Part 141, and they have guidelines establishing test procedures for analysis of pollutants as well. The 136 is your wastewater and 141 is your drinking water. There's also MPDS permit program, which addresses water pollution by regulating point sources that discharge pollutants to the waters in the United States. And this is sort of like if anybody's using any kind of water in a process, they have to treat that and release that back to the way that they found it. And these methods basically kind of follow a wastewater, which would again be that Part 136. There's, again, a little bit more in depth with this MPDS permit. There's the water pollution regulatory source, and then they have a specific website with info. And then there's the protection of environment with Part 122. Also, there is the ASTM Committee D19, and they do the study of water and promotion and standardization of terminology methods. They have water in this, which follows up with ammonia. Also, cyanides fall into this. And then total organic carbon also falls under D19. And they have the different methods that follow. Then there's also some standard methods as well that are used. And those are the 4500 series that list out the different nutrients. There's also ISO methods, the International Organization for Standardization, and those follow some of the cyanides where you have your free cyanide and your total cyanide. MBAS, also there's nitrate, nitrite in milk as well. And that is whey analysis, also baby formula. They're monitoring for nitrates in those. So some of the big purpose of testing these methods, Really big is ammonia, nitrogen, and water. Ammonia concentrations are normally low in groundwater. It can occur naturally from organic decay and waste material. Its higher concentrations are in sewage and fertilizer contamination. And you'll see that a lot with the fertilizer contamination within industries, and along also to where they drain into rivers and such, and into, say, for instance, the Gulf of Mexico. And I have a slide talking a little bit about that. There's also TKN. That's the measure of organic bound nitrogen plus ammonia. The high organic nitrogen content, again, indicates the fertilizer or sewage contamination. So you see a lot of TKN analysis in wastewater. That's also one of the most popular chemistries, the ammonia TKN combination that we offer by gas diffusion. That's requested quite a bit, along with total cyanide, and I have some information on that as well. Nitrate nitrogen and nitrate plus nitrate nitrogen is also very big. Again, those are high levels indicated with sewage, fertilizer, and industrial contamination. Then you have your orthophosphorus, which is the most common form of phosphorus in water. And again, you will see this a lot in sewage, fertilizer, and industrial contamination. There's the total phosphorus. That is a measure of both ortho plus organic and inorganic bound phosphorus. And again, these are mostly coming from man-made compounds, detergents, oil field chemicals. Also, it's natural occurring as well, as some as nitrogen, as a natural occurring mineral. Another is the EPA series 4200 for phenols. It's an ingredient in disinfectants in drinking water. There's low-level phenolic concentrations. They can impart a foul taste or odor in your water, especially upon chlorination. Then also, too, another big application that we see for low level is the BPA testing. Bisphenol A is chemical of concern found in polycarbonate consumer products such as plastic, food, and beverage containers. And we've sold quite a few of those systems doing this low-level inline phenol distillation. So here's some of the most common pollutants in ground and drinking water. The pollutants in ground and drinking water is a depletion of groundwater resources, and continued decline in overall groundwater quality is a cause of concern because of large human population around the world uses groundwater as a source for their drinking water. Studies have documented the incidence of groundwater pollution from various anthropogenic activities, such as the improper discharge of industrial and municipal wastewater, which kind of falls under those MPDS permits, application of recycled wastewater for irrigation purposes, mobilization facilitated by subsurface geochemistry, and seasonal changes, often making groundwater vulnerable to a variety of pollution from organic and inorganic contaminants. And you'll see that with the seasonal changes, especially during the warmer months, when you can see cyanogens developing in bodies of water, the phosphorus and nitrogen with the heat. You'll see your ponds or lakes or so kind of gloss over with algae and such, and those are oxygen depleting, which leads to fish kills. So here's a little bit what I mentioned about now with this is the release of nutrients have resulted in large dead zones, which you'll see from the Mississippi River Basin, which you see quite a few rivers flow into the Mississippi River, which then goes into the Gulf of Mexico. And then you have the hypoxia. And you'll see red tides, you'll see fish kills, you'll also see the reefs kind of being bleached and such, where coral reefs are being destroyed. So there is the monitoring of nitrogen and phosphorus, ammonia, TKN, orthophos, and total phos. And nitrogen and phosphorus account for over 80% of the analytes measured by automated chemistry analyzers. And well over half of these analyses are for simple nutrients of ammonia, nitrate, nitrite, and phosphate. The remainder is total nitrogen phosphorus, either by Kaldol or persulfate digestions. With the phosphorus in your environment, you'll see that in plants and also from fertilizers. It is phosphate that's consumed by algae and the potential to cause the algal blooms. Measurement of total phosphorus is important because it's with time convert to bioavailable soluble phosphate. And as I mentioned, you'll see dishwashing, laundry, and many hand detergents and soap contain phosphorus. And this content in detergent can be as high as 8.7%. And its use is to improve the quality, especially in waters that contain high amounts of calcium and magnesium. Nitrogen is an essential nutrient for plant growth, ranking only behind carbon, hydrogen, and oxygen in total quality needed. And again, you'll see nitrogen in fertilizers, which largely comes from a chemical reaction between nitrogen and hydrogen gas to form ammonia. That ammonia then can be oxidized to form nitrate. And then the fertilizers will often contain ammonia, nitrate, and or urea as the sources of nitrogen. Organic matter referred to as humic matter, manure, mulch, etc., contains about 5% nitrogen. And this nitrogen is slowly converted to ammonia by bacteria and is eventually oxidized to nitrate, then nitrite. During storm events and during irrigation, the nitrite in soil is easily leached into the runoff. One of the big things of monitoring for nitrates and nitrites is its water-soluble molecule made of nitrogen and oxygen, and it's formed when nitrogen from ammonia or another source combines with oxygenated water. And this is also a health concern where it could be involved with blue baby syndrome as nitrate changes to nitrite in their stomachs. Pregnant women should not drink if contaminated. It's more susceptible to the methylomoglobinemia. People with heart or lung disease, inherited enzyme defects, or cancer are more sensitive to this. This also causes brown blood disease in fish. And here in the United States, you see most of the municipalities have to measure for nitrates, again, for this. And you'll see that like in your consumer confidence report that comes like in your annual bill from your municipality. So that's again why it's really monitored, and also monitored in the baby formulas. So drinking water facilities are treating surface water to potable water quality, and they need to test for these following contaminants. There's free cyanide, your nitrate, nitrite nitrogen, your nitrite nitrogen, and also your orthophosphate following EPA methods 1677, 353.2, 353.2 without the cadmium reduction column for nitrite, and then 365.1 for orthophosphate. We have plenty of methods to monitor pollutants as well. Here is the ammonia nitrogen phenate. And with our products, we have these method abstracts that kind of go over a little bit with regards to the methodology it follows, a little synopsis of what's happening. So this is in regards to EPA method 350.1, or as I mentioned, we also follow some standard methods and also ASTM methods. And so we'll list out that standard method 4500 NH3H. We'll give a little bit of the performance of the method, range, rate per hour. And then also we'll mention too if we use flow injection or segmented flow. And the FS3700, which we'll talk about a little bit further in the presentation, can offer both techniques. Also, we provide information with regards to running the sample at 0.01 ppb, 5 ppb, and 1 ppb, and a little bit of the graph and results of the calibration curve. So here's ammonia TKM by gas diffusion, very popular, as I mentioned. You get to remove the distillation step from this method. This was approved back in 2012 with the US EPA. So you use a gas diffusion module for this and a membrane. And this follows with your TKN 351.2, and then your ammonia follows 350.1. And again, we provide information with regards to running the sample for you. We mentioned about the digestion of the TKN samples. You still have to do the digestion step, but again, the distillation step is removed. We have cyanides, which are very popular. This is ASTM method D6888. This is a very good cyanide method if you feel your samples might have sulfides in them. We add an additional ligand to this, and it can help dissipate sulfide at 50 milligrams per liter. Anything over 50 milligrams per liter, you'll have to dilute the sample. And there's also ASTM D7365. That's a really good method that talks about the sample preparation and how to dilute samples should you need to do that. And also, again, information we provide with each. One of the very first methods of cyanide that was approved with gas diffusion was OI 1677 for available cyanide. And again, this was the very first method that removed distillation. So OI 1677 and D6888 mimic each other almost identically. D6888 adds that one reagent, though, to help dissipate sulfide. Then in 2012, total cyanide was approved by ASTM method D75. And this is the distillation-free cyanide as well. For many years, you still had to perform the distillation step. And we do provide that via EPA method 335.4 if distillation's kind of still required. We do have a method for that. That's measured by photometric detection. And these cyanide methods, OA 1677, D6888, and D7511 use ampermetric detection for these. We have your nitrate nitrite by EPA method 353.2. And you can do this either by segmented flow or flow injection. Now, also with nitrate nitrite, we do use ISFA, and that's injected segmented flow analysis. And that's required as your sample goes through the cadmium coil. You need sort of that vigorous movement through the coil to help with the analysis and the breaking down of the nitrite to measure your nitrate. And here's examples of where we use the cadmium coil. And then when you want to do the nitrite, nitrogen by itself, there is no cadmium coil with that application. Phenol inline distillation, as mentioned with regards to the low-level analysis. This is the low-level analysis, especially for BPA. This uses a distillation module, and the phenol is distilled inline from an acidic solution, and reacts with 4-AAP and an alkaline ferrocyanide to form a red complex, and then that's measured. We also have post-distillation where you can do the distillation and then run the instrument. And that follows EPA method 420.4. So we offer both ways to do phenols. Phosphate all forms. We can follow 365.1 with flow injection, and we can follow 365.4 with segmented flow. Also, this follows standard method 4500 PG, and also an ISO method as well for the international standard. The phosphate all forms will do both the orthophosphate and total phosphate. Then we also have a phosphate all forms low level, and that's been used with municipalities that have to make sure that they are monitoring the phosphate at extremely low levels. You'll see this mostly with those that have to recycle their water. Clark County, Nevada, which Las Vegas is in, they use this method pretty extensively because they recycle their water continuously and have to measure for this. Excuse me. We also have developed total dissolved nitrogen, where we are doing inline dissolved nitrogen, and we're using UV digestion with our UV lamp. And we're using the same technology with UV digestion as we do with total cyanide. The total cyanide uses UV digestion to break down those bonds, same as with the total dissolved nitrogen. And this is also following EPA method 353.2. This is really, really good for your surface waters, domestic and industrial wastes. Then we have total phosphorus as well by inline digestion and flow injection, again using the UV persulfate method for that. So we'll touch upon the OI Analytical Xylem technology. So what we have is our model FS3700 automated chemistry analyzer. It's an advanced continuous flow analyzer designed to improve laboratory productivity by automating wet chemistry test procedures. It offers flexibility and analytical capability to meet clients' needs. It's got plug and play technology. You can pull out the chemistry plates. You can swap out the detectors. Also, flow injection valves can be added easily. If running segmented flow and need flow injection, you can add a valve quickly as well. It offers high sample throughput with low cost of ownership with microflow, robust technologies to meet the demands of every type of sample matrix. We have excellent position in accuracy. Then also to regulatory compliance, which is always important, from ASTM, US EPA, standard methods, and ISO. Some of the neat features that we added to the FS3700 is the automated injection valve, the FIA valves installed to the analysis module chassis when required to run flow injection. It's an 8-port valve. Again, it's pretty easy to add if necessary. Each chemistry cartridge is pre-assembled with all components needed to perform a validated analysis method. We just attach the pump tubing and the detector flow cell and reagent and can start monitoring. By ordering the channel, it provides a convenient way to configure the 3700, again, with modular, flexible hardware. It provides a great platform for research, in-house, or proprietary methods. We have the inline devices for reactions requiring heating or UV digestion, and they're mounted on the underside of the chemistry cartridges. The FlowView software provides user programmable control of the UV lamp and the cartridge heater set, and you can change those in 1 degree C increments. We have the photometric and ambiental detector modules that plug into the FS3700 to support methods employing colorimetric chemistries or the ampermetric measurements via cyanide. So for years, laboratories have turned to us for accurate, reliable, continuous flow analyzers. And the flow solution is our next generation automated chemistry analyzer. It's about our 4th unit that we have. It's built incorporating the AlpChem PerStorp line, which we purchased back in 1996. Incorporates some of the CN solution, the 3100 and the Flow4, our previous models, and also new technology with it as well. We have the ability to run segmented or flow injection. We have WinFlow software that's a user-friendly interface. It's compatible with Windows 10, 32, or 64-bit. And it can connect via USB cable to a computer or a laptop. It's laid out with the precision pump, chemistry plates, and then our detectors. So as kind of touched on a little bit earlier, we do have many chemistries available to provide with the unit following methodologies, cyanides, which we're well known for. Again, the ammonia TKM by gas diffusion, ammonia phenate, your nitrate nitrites, your phosphate, all forms with low level, your phenols. We can do chlorides, sulfates. We can do the cyanides by the ISO methods and the post-distillation cyanide, the 335.4. And then we have the total nitrogen and total phosphorus. The chemistries released for the FS3700s are the best methods selected from all configurations developed from previous generations. So that can be with segmented flow or flow injection, or also to if need something that has to be reconfigured or so by the injection technique, we can validate methods to say, for instance, if it's doing flow injection to a segmented flow or vice versa. The best method was selected for use on the 3700. And again, as mentioned, we can customize those. They're very modular and flexible platform. We offer 2 auto samplers with the unit, either a 180 position or a 360 position plus. With the 180 position, that's kind of the most standard one. You get 2 90 position sample racks or 2 60 position. We have 10 position standard racks for standards, wrench station, onboard peristaltic pump, and sample probe kit that works with the Z drive. We utilize the ISBTech 24 channel pump. With 24 channels, you can plumb up multiple chemistries and have those ready to go. If you have multiple chemistries to change up the plate, you have your pump tube set up, just add reagents, and away you can go with running multiple chemistries. It is a 2-channel system, so you can run 2 channels and then add additional chemistry plates by removing one and adding the other. With our platen, we have the platen extender. Pump tube is then inserted into that. We have the tension lever as well. Offer many pump tubes, and then also those are dependent upon the internal diameter and also the flow rates, and so have those laid out for each chemistry. And here's an example of what you would get with ammonia TKN. We do sell the pump tubes in 12 packs. We also have TK tube kits specific to the chemistry. And then we also have individual pump tube assemblies for each piece. So with ammonia TKN, for instance, you need an orange green for air injection. You need gray gray. You need yellow yellow for your sample line pump. The black black is your debubbler pole, since ammonia TKN in this particular instance is run via segmented flow. And then also we incorporate orange yellow. So what's in a channel? Everything needed to perform analysis is included in a channel. So if it requires flow injection, you get the flow or the via valve, chemistry plate, and your detector, ampermetric or photometric, depending upon the application. The cartridges includes the gas diffusion manifolds if necessary, heaters or UV lamps as needed, all your tubing, pump tubing, and the chemistry kit along with the full method. So here's a layout of a colorimetric chemistry. So we have the FIA valve, that's an 8-port valve. There's your chemistry plate with the mixing coils. Also showing the debubbler that's used with segmented flow. And then there's your photometric detector, which includes the filter specific to the chemistry you're running. And then what's included in an ampermetric chemistry, FIA, flow injection, is used with our cyanide methods. There's the chemistry plate with mixing teas. There's the gas diffusion module used for your cyanides, which again removes the distillation step. Then there's the ampermetric flow cell with the reference electrode. With our ampermetric detector, and this again is cyanide specific, it's one of the best ampermetric detectors on the market, and with 3700 we hone that to further reduce noise. It's not a third-party detector, it's our own. You have improved cell design and upgraded electronics. That's made up of a reference electrodes, red, working white, and counter electrode black that closes that loop. With our photometric detector, we've called that our expanded range detector for many, many years. It offers a 10x increase in signal to noise for some photometric methods. We've improved detection limits from our prior models. We can get MDLs down to low ppb. Limitation now is the flow noise. We don't have a need for auto dilution since we have such a wide linear range, but we are launching auto diluter this year, second quarter. It will be our ML600. And as I mentioned, with the wide range capability, this is a benefit using the detector in its inherently higher sensitivity. Because of that sensitivity, it can detect very small changes in the signal without increasing the gain. And this allows the detection of very low concentrations. Because the gain is not increased, it can also detect high level peaks with extreme accuracy in the same run without topping out. So here's a little breakout of the photometric detector. We have a flow cell, 5 millimeter. We do also offer 10, 15, and 20. The 20 is used for the low level, higher sensitivity methods. Sample stream flows through the detector required for chemistry. As the sample flows through, you'll see a peak rise on the graph through the FlowView software. After the sample passes through, peak will settle back to baseline until the next sample begins to flow through the flow cell. Peak is compared to the calibration curve, and a result is calculated against that curve. Here's a little bit further breakout of our detector, again with the lamp, easily replaceable photodiode lamp. Flow cells, I mentioned 5, 10, 15, and 20 millimeter available. The nanometer housing for the filter is right directly under the plate and easy to access. And so again, with the photometric detector, this is where everything coalesces and the results of the sample collection, reagent preparation, instrument operation come together to produce the final analytical conclusion. With the expanded range detection with our photometric detector, we did do some improved signal to noise ratio and increased sensitivity. And we found that with an 8-port valve plumbed with a bypass loop equal to the size of the injection loop while in load position, it alleviated pressure differential and reduced detector noise related to the valve actuation. So we're able to reduce that. We had previously used a 10-port valve, and through this work with that, found that the 8-port valve was a better solution with the Flow 3700. So we'll go real quick of the FlowView software. Real easy layout of the home screen. You start with configuring your analyzer, building a method, build a sample table, run samples, view results, and perform maintenance. It's very intuitive interface, 62 and 32-bit. Can run on Windows 7, 8, or 10 Pro. Most everybody's using 10 now. Can perform dynamic auto scaling, keeping analyte peaks on scale over large dynamic range, as I mentioned, 3 to 4 orders of magnitude. It allows you to view and edit results in real time. There's LIMS import-export configuration. You can reprioritize or edit the sample table on the fly during analysis as well. So you can add emergency samples, or if the sample comes in, you have to do something immediately, you can add that to your run. The software begins with the launcher, FlowView launcher, easy to configure, launch icons for each 3700 system and provide access to FlowView software and the system utilities. You can add a 3700 to that and give the instrument appropriate name. That's typically the serial number of the unit, and then select your COM port. The configure analyzer gives you the ability to set up your pump. In each method, we provide the pump run speed as well. And you can choose if you have valves to enable or disable the valve. The channel, this is to enable, disable the channels, cycle duration, and channel name. So you can pick the chemistry that you are running. It also has the heater enable, disable. You can configure the detectors from here. If you're running a method that's cyanide, that's when you can drop down and choose the ambimetric, or a colorimetric, that's the photometric. And also that's where you can also then set up your sampler, the 180 or the 360 positions setup. Next, you would build your method. It's divided into 4 sections that allow the user to define the method and the calibration. We have a calibration table, the actual calibration, your signals, peak marking, and then quality control. With the calibration table and calibration, define the concentration and ID for all the calibration standards. You can highlight a channel, then choose a curve fitting method and concentration units. With the signal peak marking and quality control, the configure peak quantitation, height and area, baseline handling, smoothing, and seek peak identification parameters. And in the quality control, you can set that up for up to 13 different quality control samples. Then we have the build sample table. This screen allows the user to find the sequence of samples to be analyzed. And then finally run samples. Open the run sample screen to control the sample sequence and monitor data as it's collected. The FlowView channel display options, there's multiple ways that you can view. You can see your channels, channel 1, channel 2. To view one channel, press the channel number button to display that individual channel's results and detector signal in the viewing area. You can also view calibration curve for a channel during a run by pressing the view calibration. Here as in view results, you can find the curve equation and calculated value for the standard. Then there's the press all channels button to display a split screen view of detector signals and sample peaks. There's the view results screen that you can open to view results. Results may be reviewed from an active or completed sequence. And here's like a basic layout of the view results. There's the configuration, the method. This tab is the calibration results. There's your sample table to choose from. And then there's the channel 1 or channel 2, which signifies what's when you were running. You can also then export your results. Press the export results button. Open a dialog to select what information and file format you wish to save the results for the active channel. And then you can export those to those. And here's like an example of the export results. You can choose different peak data parameters, header information, peak graph, peak data, calibration graph, calibration data. We also have incorporated a maintenance screen as well. And this allows the user to view information and test the operation of all components attached to the Flow Solution 3700. Also, there's a self-test. This is a very good screen to do troubleshooting from. It displays a window to initiate a self-test of the FS3700. You click start, the test runs continually, run until you press the stop button. Heaters are turned on and off, set to a predetermined set point, valves, switch, communication to the pump and sampler checked. In addition, the detector signals are monitored as well. Some additional features is a sample table import from CSV file from Excel. It's a full screen GUI, ability to pause and resume the run between samples, emergency sample capability, and it also supports multiple languages, English, Chinese, French, and Spanish. And then a quick overview of the library, again, of our methods. We have the cyanide methods that can do the ability to remove the distillation step with D6888, D7511 or A1677, and we also offer EPA 335.4 for the post-distillation cyanide. Those methods employ flow injection analysis. And again, what's very unique about our 3700 is you can do simultaneous available cyanide and total cyanide. You can also run a channel with flow injection, a channel with segmented flow. Then we have the colorimetric analyses, your ammonia TKNs, your nitrate nitrites. Those are combined is because you can run those simultaneously as well. Your phosphorus, low-level phosphorus, your TKNs can be run together with the phosphorus since you have to do the digestion step still. We have the phenols and then chloride. We also have sulfate by 375.4. And then also the total nitrogen and total phosphorus by UV digestion. We also have some other methods as well. And so you can acquire with your resources with Giovanna or our friends here with Analytech on different methods and such. We have the method abstracts available for folks to review. And so that concludes presentation. As I mentioned, we are working on a auto diluter, the ML600. It's going to be great for folks that can make up your standards and such, and then also do dilutions to your samples as well. And that is in beta and should be launched hopefully at the end of Q2. So if there's any questions or so, happy to answer. And if I can't, we'll get an answer for you and get that back to you.
Okay, thank you very much, Hank, for this presentation. So I'm going to say to the audience that we are in the answers and questions part. Bueno, entonces esto ha sido toda la presentación. Pasamos a la sección de preguntas y respuestas. Si tienen alguna pregunta, por favor háganosla llegar al chat o a la sección de preguntas y respuestas. Puede ser en español o en inglés, como ustedes lo prefieran. Y en unos momentos estaremos dando respuesta a sus preguntas.
Okay, in chat, see a question about some of the analytes you mentioned to be done by IC What is the difference versus FIA and segmented flow? Very good question. Flow injection with the FIA valve is a continuous flow. So you're going to have sample reagent mixed, and it's going to be continuously flowing to the detector. We recommend, if people are new to automated chemistry analysis, that flow injection is a little easier to start out with. Segmented flow, it inserts bubbles, and so that gives you dispersion. So when I mentioned a little bit about the ISFA and segmented flow, you have sample, then you have a bubble, and so those spin through the pump tube and you're getting very good dispersion. So segmented flow is good for low level detection because you're going to have a thorough run through the pump tubes into the flow cell. With a flow injection, the sample is a continuous flow, so you don't have a full and complete run through there. So your peaks are going to be a little bit different from the shape. Flow injection is, again, used a little bit more for sample throughput if you have a large sample load with the continuous flow through there. And again, segmented flow is kind of good for low-level detection because you're getting that complete reaction with that. So with flow injection, that's used with cyanide. You can also do that with the nitrate nitrite. Why we use it with total cyanide is because you need to have dispersion as you're breaking down those bonds of the cyanide ion. And so total cyanide needs to be really, really vigorously digested with the UV digester lamp. And then we also have dispersion throughout that. Okay, I need help. And versus IC, we haven't really had any kind of differences or run with an IC. I know prior latchet instruments, you could have IC with their instrument. So I apologize, I really don't have much knowledge of ion chromatography. I know that you could choose between that with the latchet, but with us, it's the flow injection or segmented flow. I know that like nitrate nitrite, I know people bring up ammonia and such, and so the ammonia can be run by gas diffusion or also by the ammonia phenate. Nitrate nitrite goes through a cadmium coil instead of a column on the IC, and that's about basically I know, so I apologize, I'm not very familiar with ion chromatography.
Okay, thank you, Hank. We have another question from Melesio Juarez. He's asking about which method do you use to prevent the contamination in base of these components, in base of this presentation. Just let me ask to Melesio. No alcanzamos a entender muy bien la pregunta. ¿A qué tipo de contaminación se refiere? ¿Se refiere a la comparación entre FIA y SFA o a contaminación como se habló en la presentación? O sea, referente a la contaminación por fósforo, por nitrógeno, etcétera. No sé muy bien a cuál de las dos cosas se refiera. Si nos pudiera escribir por ahí. o se está refiriendo a contaminación cruzada entre muestras que pudiera darse, por ejemplo, en un análisis de flujo continuo. Si se encuentra por ahí todavía presente Melesio Juárez, que nos pudiera, por favor, aclarar esa parte de su pregunta. Ok. Está bien. Gracias, Melesio. Okay, Hank, about the first question of Melesio, and it's about the contamination that is probably to have in a FIA analysis. Which method could be better to prevent this contamination, the FIA or the SFA?
Possibly with segmented flow, again, because you have a complete reaction with that. Also, you can insert a blank in between runs to help flush the lines. I know it's not normally brought up, and we discussed that with the product specialist one time. Because I know, like say with TOC, you can run a blank to flush the line and such before then, like another samples, like if there's going to be contamination and such. So you could put a blank in there. Also, too, after say, like for instance, any kind of run, you can then flush the lines to prevent any kind of cross-contamination. So possibly SFA, again, because of complete reaction. And then, as mentioned, you can insert a blank if necessary as well.
Okay, thank you, Hank. Hank nos comenta que sería mejor el análisis por flujo segmentado, ya que la reacción, digamos, se lleva entre esas burbujas de aire que permiten que no haya una contaminación, digamos, como se pudiera dar en el análisis de flujo continuo, lo que sería el FIA. También nos sugiere, recomienda correr algún blanco entre muestras para prevenir ese tipo de contaminaciones. And another question, Hank. It's more about the environmental contamination and the pollution of the water, the air, the soil. And what do you suggest to the society to have an unhealthy environmental, an unhealthy and self environment, and for all the living I don't, excuse me, for all the living people there. What do you suggest to the people to preserve the environment, to pollute the less possible?
With samples, if there can be again, back to to possible contamination and such, samples can be filtered as well. Since this is macro flow, microflow, you don't want to have kind of like any kind of things floating through the sample and such. So those can be filtered, or also too, they're going to be digested or distilled as well in the sample preparation step before analysis. Also too, there's the different regulatory or kind of methods that have been written, especially with the permits and such. So kind of essential that there is good kind of regular testing and such of especially your surface waters and such that are going to be used for your drinking water. And so that's why there's like the Clean Water Act that was kind of set up back in the mid 80s. And then also too, you have the MPDS permits, and you have that 40 part 136 CFR for the regulatory. So in case people aren't following like EPA methods, that's why there's the standard methods and also the ISO methods and ASTM methods as well. So there's multiple methods that can be used to run these analytes. And at those websites, they have it all broken out as well. And so they have a columns that'll show which ones can be used. So in case like EPA is not used or so, there's the other methods, and they'll be kind of with your specific analyte, and then which regulatory body you can use.
Okay, great. Thank you very much, Hank.
Yes, sir.
Y bueno, Hank nos comenta que respecto a la contaminación del ambiente que se puede dar debido a este tipo de análisis, bueno, pues él nos menciona que hay distintos métodos, hay varios métodos que se pueden utilizar para analizar una cierta especie química, por ejemplo. Sin embargo, pues hay que optar por los que presenten un menor impacto al ambiente, incluyendo desde la preparación de la muestra, el manejo de la muestra, hay tal vez algunos pasos en los que se tengan que utilizar ciertos reactivos, dependiendo si se sigue cierta metodología o no. Entonces, lo que hay que cuidar pues es buscar siempre la metodología que genere un menor impacto al ambiente. Bueno, no sé si haya más preguntas por aquí en el chat o en la sección de preguntas y respuestas. Bueno, me parece que ya no hay más preguntas y respuestas. En dado caso de que quedara alguna duda, pueden escribirnos. Por favor, mándenos un correo con cualquier duda que tengan y nosotros buscaremos resolverla. Como comentaba Hank en la presentación, también por parte de OI Analytical y de Analytech Industrial and Research, podemos proveerles información más a detalle de todos los métodos de análisis de los que Hank nos estuvo comentando durante esta presentación. Lo que se refiere a análisis de cianuros, de nitrógeno, de fósforo, sea cual sea su requerimiento, por ahí podemos proporcionarles información más a detalle sobre la metodología que sea de su interés. Y bueno, si ya no hay más preguntas, más comentarios, solo me queda darles las gracias por su asistencia a nombre de OI Analytical y Analytech Industrial and Research. Recordándoles también que este seminario va a estar disponible en el canal de YouTube por si lo requieren volver a ver, por ahí si lo pueden compartir entre sus contactos a alguien que sea de interés. Bueno, eso sería todo por nuestra parte. Si no hay ninguna duda, no me queda más que desearles un excelente día. Muchas gracias. So Hank, it seems like we don't have more questions.
Okay.
So I think this is the end of the presentation.
Right.
I want to thank you to you very much for your support. And I don't know if you have another comment to say.
Oh, just thank you for the opportunity to present. And if you have any questions or need any further assistance, please feel free. You can reach out to your colleagues here, and more than happy to help or supply information. So thank you again very much for the opportunity to present our FS 3700.
Okay, thank you very much, Hank.